It's great to be here, and I really appreciate the invitation by Micronutrients to come out here and share some of our work. And so as Scott indicated, the title of my talk, what I'm going to talk about is gut health and what dictates it. And this is a pretty big question, and I think we're all still kind of trying to understand, well, what is gut health? And how do you measure it? And I'm not going to say that I'm going to have the solution by the end of this talk, that we're going to know exactly what gut health is, what the biomarker is. But hopefully, some of the data that I present is going to shed light onto what aspects of gut health we need to be focusing on, and are there potential biomarkers? Let's see if I can, that we can use to assess gut health. And another topic of this talk is what is going to dictate gut health? What factors in the production environment are going to compromise GI health and function? And I'll share some of our work, mostly from a pig model, but once we get past the rumen, you see the responses to what we're studying, which is particularly stress response, is very similar across species. So I think that we can translate what we're learning from the pig to multiple species. So one way to try to define gut health is maybe looking at the critical functions that the gut does on a daily basis. And Dr. Penner did a really great job setting this up talking about different aspects of gut functions and what the gut requires to do. So this is a cartoon here of the intestinal epithelium in the small intestine that lines basically this single layer of epithelial cells. And this epithelium serves many critical functions. On one hand, it must digest and absorb nutrients. It must transfer a tremendous amount of water and fluid across the intestinal epithelium, which is critical for digestion. And it must also reclaim this water, and that's mediated mainly by its ability to transport these electrolytes. And this is also critical mechanisms by which nutrients are absorbed across the gut. Another major function, again, covered before, is barrier function. I'm going to talk a little bit more about this, how stress impacts this critical barrier in the gut, and what factors are dictating this. And again, just to review Dr. Penner's talk, there's a lot of different aspects of barrier function. There's the tight junctions, which regulate the permeability of the epithelium, as well as all these other factors, the microbiome and extrinsic factors like mucus and protective antimicrobial peptides, which serve as this critical barrier between the body and the outside world, which in the lumen contains a massive amount of antigens and toxins. So its job of this epithelium is to selectively exclude potential antigens and toxins from getting across this epithelium. Another major function of the gut is immune function, and we know that the gut represents the largest immune organ in the body. So most of the immune system is concentrated in the gut. And this is critical from a defense point of view. This gut must have an active immune system that must, one, allow it to selectively regulate or respond to commensal microbiome, but at the same time be able to detect pathogenic processes and be able to respond rapidly and prevent the spread of infection. And another component, if you look at what the intestinal immune system is composed of, you have secretory products and immune cells. But we also have this enteric nervous system, which we know that the intestinal or the gut mucosa and submucosa contain just as many neurons as the spinal cord. So it's one of the most richly innervated organs. And why is this important? Is because this entire nervous system has tremendous control over the immune system in the gut, as well as transport and barrier function. So a lot of the stressors and factors in our production environment can change either the development or activity of the nervous system, which leads to pretty profound changes in the gut function in general. Okay, so we also know that the gut and the gut function or the gut epithelium, what happens in the gut is not really restricted to the gut, because the gut communicates with many different systemic tissues, including interactions with the nervous system and neurohormonal responses and immune responses that communicate with the brain. This is the brain-gut axis. We know what happens in the gut can change behavior and vice versa. What psychological stressors can have a rapid, pretty significant effect on the gut as well. And not only in the central nervous system, but every other organ has been linked to changes in gut function. So this is a common theme where a lot of conditions, whether they're lung infections or changes or systemic diseases, the gut is a common comorbidity where there's the gut is either setting up the whole body for responses and disease susceptibility in other organs as well. So that's why understanding how the gut works and its gut function is really critical even beyond GI diseases. So what are the factors that impact gut health? And it's really complex. And Dr. Havertine and Dr. Penner both covered focus on this aspect. On it. It really depends, because we've known a lot that factors like stress, age, the biological sex of the animal, and environmental factors such as diet, genetics, pathogen load, these can all have an effect. However, what's really critical is the interactions between these effects, because we know that any one of these may have an effect, but it may not be enough to influence the disease process. Animals respond to stress all the time. It doesn't mean that we're going to see changes in performance. But it's really what becomes critical is all these other interactions, the age of the animal, the sex of the animal, which is a particular focus in my research laboratory. I'm going to share how these factors interact to influence and shape gut health. Now, I talked about the changes in age and the interactions between ages and the impact on gut health. And this becomes really critical when we're thinking about the early development of the GI tract. We have, during the first several weeks or several months of life in animals, there is this tremendous development and maturation of the gastrointestinal system, including the immune system, the epithelial barrier and transport functions, as well as the enteric nervous system and the microbiome as well. And this is a very critical period, and it's a period of plasticity, meaning that how the gut develops during these first several months of life is going to shape how that gut will respond and develop throughout the remainder of the lifespan of the animal, or production lifespan. So this is termed the critical period of gut development. And in nature, this is really meant to be a very quiescent period where there's very limited activity. The immune system, multiple mechanisms, which the immune system is dampened. And this is just meant to be a very quiet period in development. But in production, this is the time when some of the most severe stressors are imposed on animals. And whether it's in the pig, there's a lot of, or calf, this is where a lot of management practices and stressors occur, and it's occurring in the peak period of gastrointestinal development. And this, because of the plasticity in this environment here in the gut, this has potential to alter the long-term development and the function. And that's what we're really focused on. Or what are these early life factors and what are these changes that stress can have on the early development gut that are going to shape how that gut will function throughout the remainder of the lifespan. And then how this is correlated with disease risk. And we know that animals, from a clinical perspective, animals get an infection or they're challenged early in life. Those animals never go on to become full, reaching their genetic potential. We've known this for a long time. And this concept is not new, because even if you look in the human literature, the effects of stress on early childhood, the sources of this adversity or stress, also lead to this increased disease trajectory as well. So it's shown over many species, including humans. So we know this critical period when there's changes to the or trauma to the gastrointestinal tract or injury leads to this lifelong trajectory for impaired function. So we study weaning stress in piglets, which is arguably the most stressful period in pig production. And you can see here, these are animals. They're about 2 to 3 weeks of age. They're with their mom and litter mates. And at this critical period in gut development, we wean them. And we put them in environments where there's exposed to several stressors all at once, including maternal separation, changes in social hierarchy, changes in environment, pathogens, diet. There's a lot of things going on that are a tremendous stress during this critical period. And GI disease is one of the most first and the most common causes or result of weaning stress in animals. But you can apply this to any other species as well. Weaning is a little bit different say in the dairy calf, which is even earlier, with 1 day of age, or beef cattle. This is a very stressful time for animals, and it's occurring during peak developmental time. So if you look at where weaning occurs in nature, and I'm not sure why this is working, it's really quite a difference. You know, in nature, especially in the pig, the pig will wean themselves about 3 to 4 months of age. It's a very gradual process, but in the production environment, this is occurring between 2 to 3 weeks of age. So again, it just highlights how different really it is in respect to nature. And also in nature, this is what, in the pig intestine, we know GI development kind of plateaus and ends at about 3 to 4 months of age. And this is the natural time when the animal is going to wean themselves in a natural environment. So we're really imposing a lot of stress really early during this developmental phase. And we've known for some time that weaning and weaning age, looking at weaning age in the effects on long-term production are quite significant. This is a study by Dave Rosero and Dean Boyd at Hanner System looking at incremental increases in weaning age and looking at lifetime average daily gains, so from wean to marketing. And you can also see the wean to finish mortality. So we see these benefits of changing and influencing stressors such as wean age throughout the whole life, you know, of these animals. The problem is that many of these production practices, such as managing weaning, are inherent parts of the production process. We can't really manage wean age. So we really do need to gain a deeper understanding of what the underlying mechanisms are that are driving some of these increases in disease susceptibility. So this is, again, the focus of our laboratory. We're trying to understand what are these factors early in life that are driving these long-term increases in disease risk, and trying to understand what's going on at the gut level. So one thing we're interesting in the gut barrier development in the animal. And again, we already overviewed some of what's the importance of the intestinal barrier to health of the animal. And it consists in the intestines, a single layer of epithelial cells that provides a barrier between the outside world and the body. And again, this is an overview of what this barrier function entails. But this is an extremely critical mechanism because we know that any defect in intestinal barrier function or increased intestinal permeability has been linked to a number of adverse consequences. And if you look in the animal or human literature, the breakdown in barrier function is a common inciting mechanism that predisposes people and animals to disease. So once this barrier becomes broken down, you get leaky gut. It can lead to a number of consequences in disease situations. So this is what it's so critical. So how we measure intestinal barrier function, mainly in our lab, is through an Using chamber method where we can place intestinal tissue in this Using chamber system, and this is recently isolated intestinal mucosa. So the tissue is viable in this system, and the tissue is bathed with oxygenated Ringer solution on either side, and we have electrodes attached to here so we can measure certain electrical properties across the gut. So it's a really nice sensitive method to measure intestinal barrier function and transport processes, which I'll cover in detail this morning. But one of the ways, and Dr. Penner really set this up nicely, is to measure the mannitol or the permeability flux across this epithelium, where we can apply a probe that crosses through the paracellular space through the tight junctions and measure how much of that probe gets across during a certain time. So a high flux rate is equal to an impaired barrier function or leaky gut, whereas a low flux rate would indicate an intact barrier. So one of our first questions in our experiments that we wanted to ask, well, what is the impact of weaning on intestinal permeability in pigs? And this was a study where we took pigs that were 21 days of age, and then we weaned them, and we 24 hours later, we mounted their intestine on Using chambers and looked at how permeable they were compared to their unweaned littermate controls. And as you can see, within 24 hours of weaning, you see this pretty dramatic increase in intestinal permeability, indicated by this mannitol flux across the intestinal mucosa. And we've seen this in several regions of the gut, including colon, ileum, and jejunum. So it's quite dramatic how much this gut breaks down. And this is in 24 hours, but some of our recent data suggests that this happens, the intestinal permeability can happen within hours. So within an hour, we can start beginning detecting some changes in permeability. So it's a pretty rapid process. And when you get breakdown of this gut barrier, this leads to the ability of these antigens now to freely cross the gut epithelium and create a lot of inflammatory responses in the underlying mucosa. And here's we did a transcriptome analysis, which basically looked at all the genes associated with immune regulation in the gut. And we see hundreds of genes that are rapidly upregulated in response to weaning. So it's an impaired barrier, and it's an intense inflammatory response occurring very soon after weaning and during a critical period. We also wanted to understand how different factors can impact the degree of injury at weaning, and one of them was age. Does age matter? We knew that there was a steep developmental curve as the gut develops post-weaning, even between days and weeks between animals. So we asked, well, whether or not if we delay weaning even a week to 10 days later, what is that impact on the response to this weaning stress? And what our data showed was that this is call these EWS, or early weaned animals stress. You see that this is, again, 24 hours, you see this marked increase in intestinal permeability. But animals that were weaned at a slightly or a later age, they get intestinal permeability post-weaning, but it's far less severe. So the gut is probably more able to respond to those stressors associated with weaning. And one of the questions, well, maybe these animals that were just kept with their mom, maybe they're just not as stressed and they're more able to respond to the weaning. So we looked at serum cortisol, which is a surrogate marker of the HPA axis stress response, to see if these animals were less stressed. And if you look at the cortisol responses, yeah, they're elevated in the early weaned pigs, but they're also elevated in the late weaned pigs too. So this didn't really correlate with gut function. And I think what this says is that the stress response, or the stress of weaning, is going to be stressful no matter what for the animal. But how that gut responds to this stressor based on age is very different. And so because we knew the degree of injury and intestinal permeability that's occurring during a critical period, we also wanted to know what are the long-term effects, because we know this is a very plastic period. Can we have long-term effects of intestinal permeability, or does it recover, and how long does it take to recover? So we looked at the impact of weaning age at different days post-weaning, all the way up into adulthood in pigs. And what we found was that there's, again, we see this marked increase in intestinal permeability in early weaned animals. But as you carry this out, and even into adulthood, they have this persistent intestinal permeability defect throughout into adulthood. And we've shown this also in other models, other species as well. You impose an early life stress during a critical period, you have long-term programming effects on your gut barrier. You have a persistent leaky gut throughout life. And another question that we wanted to know, and it may not be as relevant to like dairy cows, but does sex matter? Does biological sex matter? Because we know there's a huge sex bias and a lot of stress-related diseases in people, with females at higher risk to develop stress-related diseases like irritable bowel syndrome or inflammatory bowel disease. So we began to look at this, and what we found comparing male castrates to male females, this is intestinal permeability post-weaning, early, and these are the late weaned and then early weaned animals. We could see that females, animals, have a much higher gut permeability in response to early weaning stress. And this persists into adulthood. So even factors like the sex of the animal are factors that we really need to consider when we're dealing with trying to assess the impact of stress on the gut. So just to summarize our work on barrier function, we've shown that factors such as wean age and biological sex during this critical period of the development can really change the trajectory and long-term GI function. Just really emphasizing what happens early in life can really have long-lasting effects. So knowing that the importance of the intestinal barrier to function, we wanted to try to understand, well, what is driving gut permeability? What are the underlying factors? Because if we can understand what are these underlying factors, then that's maybe areas that we can develop maybe targets or therapeutics or dietary strategies that can maybe improve intestinal barrier function in stressed animals. When we're looking at regular H&E histology, or we look at the villi length and just overall structure of the gut, we never see any major differences between early weaned and late weaned pigs, despite this this increase in intestinal permeability and these functional changes. Really at normal, just assessing villi height, we really don't see anything. And we've had board, certified blinded pathologists look at these tissues, and we can't really see anything at the H&E level. But interestingly, we became interested in a cell type called the mast cell, which doesn't show up routinely on an H&E stain. And I'll tell you a little bit more about mast cells. They're innate immune cells. But if you look at early weaned animals in red, 2 months, 6 months after weaning, we have this pretty significant increase in the number of mast cells that are populating the intestinal mucosa. And also, they're pretty similar in the numbers between, say, male and female animals that are exposed to early life stress, but then also the mediators that they're being released. So we wanted to kind of understand a little bit more about what these mast cells can, what their role is in regulating permeability. And we know about mast cells from years of research. We know that they're critical innate immune cells that can play beneficial roles, including pathogen defense, wound repair, and resolution of inflammation. But they're also, when they become hyperactivated or increased in number, they can also contribute to a number of disorders. And even in humans, they've been associated with leaky gut, inflammatory responses, allergy, anaphylaxis. And mast cells are really defined or mediate their functions through their associations with other cells, because they're not a cell type that are very high in number in the gut, but through their interactions with a number of different cell types, including the epithelium, this is where they modulate their function. So they can modulate gut permeability, they can modulate inflammatory responses, and interact with the nervous system. And looking at mast cells and how they respond to an animal exposed to any stress, they're one of the first cells that are activated in the immune response. So within minutes after a stress, serum histamine levels, which is a major mediator of mast cells, become rapidly elevated within minutes after any type of stress. So this is like an important alarm system in the body that becomes activated in response to stress. So we want to try to understand what is the impact of this mast cell activity. This is just some other work just showing how early weaned animals have more increased mast cell activity soon after weaning in the gut and release of mast cell mediators. But we want to understand, well, what is the impact? What is the role of these mast cells? So we actually blocked mast cell activation by pre-treating animals with sodium cromolyn, which stabilizes mast cells, and looked at intestinal permeability. And what we see, you can see the weaned animal has a significant increase in intestinal permeability, but when we block or stabilize mast cells, we were able to significantly reduce the level of gut permeability in these animals, suggesting that mast cells are playing a critical role in modulating leaky gut in stressed animals. So, and this is just other work. We use a lot of models in my laboratory, and this is a model where these are animals that are devoid of mast cells, so the mast cell deficient animals. And when you subject them to stress, this is intestinal permeability in a wild type animal, animals without mast cells do not exhibit increased intestinal permeability. And we can actually give these mast cells back to these animals and restore intestinal permeability. And this is just to show that this is an effect that's translated across species, and it really shows the critical role that mast cells play. And then also looking at human diseases, we know that stress-related diseases like irritable bowel syndrome, which has a higher prevalence in females, they also find increased mast cells in IBS patients as well in the gut. So it looks like early life, and it's also a condition associated with early life stress as a risk factor. And we've also shown this in rodent models as well. And then just looking at currently what our lab is trying to understand now, what are the factors that control mast cell activity in the gut, and can we possibly modulate that? And we've identified several mechanisms, including the CRF system, which is a corticotropin releasing factor in regulating mast cells, as well as how being male or female can impact the reactivity of these cells in controlling gut permeability. Okay, so I talked, kind of summarized our work on the gut barrier, but now I want to, for the rest of the presentation, I wanted to share some new work that we've been doing, looking at the impacts of early life stress or early weaning on nutrient transport. It's another critical function of the gut, because we know that early life stressors can set up to poor growth performance, feed efficiency throughout the production lifespan. And we wanted to try to understand, well, also, could this be related to changes in nutrient transport function that's impacted by stress? So just a quick overview that's going to help understand some of the experiments that we actually conducted. I'm just going to focus on nutrient transport of glucose. And we know this is the basic mechanism. This is an intestinal epithelium. This is the brush border. And one of the primary routes of glucose nutrients getting across the gut is through this sodium glucose transporter. And this is an energy-dependent process that drives sodium and glucose across an electrochemical gradient. And that's how, and then glucose enters into the bloodstream through a transporter called GLUT2. But we also know that under high luminal glucose concentrations, like such as during a meal, that these GLUT2 transporters can also contribute to intestinal transport. And I'm going to tell you why that's important once we start looking at how stress affects this. And how do we measure glucose transport? Well, we actually been measuring glucose transport. One way is ex vivo, again through these Using chamber techniques, because if we can apply luminal glucose to the gut, and it will generate a charge, an electrical charge, and that's directly proportional to glucose transport across the gut. So it's a very sensitive measure on ways that we can measure glucose transport. So what did we find? We looked at the impact of early weaning stress on the development of intestinal glucose transport in the gut. And similar to other studies, we weaned pigs either at 18 days of age or 28 days of age and grew them up about 2.5 weeks post-weaning, and then looked at their transport properties across the gut. And what we found in both the jejunum and ileum was an early wean animal has significantly reduced ability to transport glucose across this sodium glucose linked transporter across the intestinal epithelium. And we've found this, carried this out even months past post-weaning. So it's a pretty pronounced and long-term effect on the intestinal epithelium. And we became kind of interested, well, could this be their impaired ability, could this be potentially related to why these animals are more prone to growth performance reductions? And so we wanted to figure out a little bit more about what's driving this reduced sodium glucose transporter. So we looked at expression of these glucose transporters in the brush border membrane. And what you can see is this is also significantly decreased, the amount of protein of these glucose transporters in the brush border. So that could in part contribute potentially to the mechanism. But interestingly, if you look at villus morphology and surface area and villus height, there's really no difference there. So this really suggests that there's potentially not so much the villi length, but the function and localization of these transporters. We also want to assess GLUT2, which is that other glucose transporter across the gut. And interestingly, we found that early weaned animals had increased expression of this GLUT2 transporter across the gut. And what does this mean functionally? Well, what we know about GLUT2 is that this is a very high, it's a low affinity but a high efficiency transporter that can transport 3 to 4 times more glucose across the gut than GLUT2. So in the early weaned animals, or animals exposed to an early life challenge, they have this shift in expression for increased GLUT2. And if you look at functionally how much glucose is getting across using labeled glucose, early wean animals actually have more glucose being able to be transported across their gut, despite having reductions in performance and growth rate. So we felt this was kind of a really interesting aspect of physiology, and really, what is the significance? And this is kind of the big question. Why would an animal that's exposed to an early life stress during time shift their transporter capabilities to actually become more efficient in transporting nutrients across the body, across the intestinal wall? And it really starts to begin to ask some questions. Well, how is this glucose utilized? What we see from unpublished data now, but we just ran a pretty big trial looking at it, and we see that there actually is more glucose getting across the gut of a stressed animal, especially an early life stressed animal, where we see higher serum glucose levels. But we also see increased glucose transporters on different tissues throughout the body, including adipocytes. And we also see evidence of increased visceral adipose tissue development, and also increased glucose transporters in immune organs and immune cells as well in these animals. So what we think is that, what could this possibly mean is that this shift in glucose transporter activity could just maybe be diverting nutrients. Maybe this is more of a partitioning effect where somehow, if you have the early life stress or early life adversity, you may become even more efficient in extracting nutrients from your environment, but these are potentially going to support the immune system and other maintenance needs of the gut, because we know from an immune system standpoint, there is a very active immune system in animals that have been stressed. So maybe by understanding what's controlling nutrient transport under stress, that we can uncover some mechanisms and maybe some targets to either nutritionally manage animals that are under stress, or potentially just understand targets that can be modulated during this process to improve nutrient transport or utilization in these animals. And we've also shown, which I do not have time to show as well, is we've also looked at similar mechanisms of amino acid transport. And it seems that under particularly early life stress conditions, they develop a different set of amino acid transporters across the gut. So we're going to be looking more into this and trying to understand how this reflects different nutrient needs in different animals, and how we should be thinking about animals that are maybe either been exposed to an early life stress, how their gut develops differently, how we can use this information to inform us potentially through different nutritional requirements. So to summarize here, the take-home point is that this early critical period in animals, the first several months of age, represent this critical period of development in the gut. But this is the time when most of our production management stressors occur and can have long-lasting impacts on gut development and increased disease trajectory. So what happens here doesn't recover. And what we've been finding out is that there's some pretty pronounced and long-term changes, including a persistent leaky gut, altered nutrient transport systems. Some data I didn't cover was immune system dysregulation, as well as changes in the whole nervous system. So essentially, stressors during times of this critical period can pretty much reprogram and map out a different function or fate on how that gut is going to develop. And where this becomes important from understanding from a practical standpoint is to really understand what about these stressors early in life that are going to change this trajectory. And some of these stressors may be manageable, some may not, and we may have to find a deeper underlying mechanism to maybe intervene here to promote a normal development of the gastrointestinal tract and improve health and performance across the lifespan. With that, there's many people involved in these projects. This is my lab, current lab, and both former and current members, and this is our funding. And with that, I'd like to take any questions.
Thank you, Dr. Moeser. We do have a few questions here. The first one is, has there been research conducted to identify why certain animals do not suffer from GI issues that occur in the rest of the population during challenge periods? So why do some respond or are affected and why some are not?
So yeah, that's a sorry about that. It's a big question. I think there are several factors. I think most of the talks this morning, or the theme was how several factors and interactions can determine how animals respond to the environment. One example, if you have groups of animals that are at different weaning ages, potentially animals that have been either exposed to a challenge earlier in life or maybe a different weaning age, that can make them, we know that can make them more susceptible. Interact that with differences potentially in maybe the biological sex of the animal and how they're going to respond to the stress is really going to come together. So there's a lot of potential factors, and it kind of makes things more complicated, unfortunately, as we learn more about all these host environment interactions. But from a research standpoint, we really do have to pay attention to these responses. I know that when we're generating data, we always have these outlier animals in our populations, and they're annoying from a statistical point of view because they may ruin your p-value, but we really do have to, if you have sufficient numbers of animals, start looking specifically at these outliers, the animals that don't get sick or the animals that do get sick, and kind of really become to pay attention to them, because I think that variation is really critical.
Yeah, well, thanks. The other question here is, do you see increased uptake of other nutrients or just glucose? Could this extra glucose be used for neutrophil function in the stressed animal?
Yeah, yeah, another great question. We've been looking at some other transporters that we've seen. Interestingly, we also see increased GLUT5, which is a fructose transporter as well. So that's another sugar that's increased. But looking at, we've also looked at amino acid transport as well, and we're still trying to understand what the differences are there. But we've seen, similar to glucose, we've seen some changes in specific amino acids being downregulated through electrogenic or sodium-dependent transport events, whereas other amino acids, particularly positively charged amino acids, are significantly upregulated. So we think that as it's not all uniform, and I think by kind of characterizing out which specific nutrients and amino acids and sugars are differentially regulated under different aspects of stress, I think we can come away with a pretty good understanding of maybe this is reflecting the different nutrient requirements or needs of the animal. And as those, the other part of that question is, yeah, could this be going to an enhanced immune response? And likely. We know that under, especially acute stressors, the immune system becomes highly activated, and part of that with the increased immune system, and you know, from Lance's work as well, an activated immune system requires a tremendous amount of energy and nutrients to facilitate, and an animal's response to stress is always going to be towards defense and survival first, and maintenance before any type of performance and growth. So those types of critical systems that come activate are going to receive priority of nutrients. So I do believe that's true. I believe we need to try to understand which nutrients are fueling potentially the nervous system and other systemic sites, and then I think that will be critical.
Thank you. Thank you very much. Let's give Dr. Moeser a round of applause.